Thank you everyone for joining in. I hope everyone can hear me. I'm going to be telling you about the work we've been doing in our lab and our clinics to do with ciliary phenotypes and autosomal dominant polycystic kidney disease. And I'll start with a little bit of introduction about the landscape of research that that we are involved in, and other labs and scientists around the world. So the research can be divided into three broad categories: lab research, often also called basic science, and that then progresses to animal research, and then human or clinical research. And these are not static individual research areas. They're all quite dynamic and interact with each other. And often a term is used, bench to bedside or bedside to bench. And you can see the bench on the extreme left and the bedside on the extreme right. Our work sort of spans all three areas, and I'll touch upon a lot of work which we've been doing from a basic science aspect. The aim of this study, which we've been doing with funding from PKD Charity, is to study the cilia phenotype in patients with autosomal dominant polycystic kidney disease, and to look at the cilia and correlate it with the disease condition itself, and ask questions as to whether the cilia can tell us anything new about this condition and how to treat it. So you all know what a polycystic kidney looks like by now, and the key question is, what makes a tube-like structure convert to a cyst? So on the extreme left panel, you can see the kidney here and a tubule and a collecting duct. Now, the cysts starts from these tubules and the collecting duct, and you can see on the extreme right that the direction of cells growing is generally top down, so forms a tube. But in cyst formation, the cells do not follow that pattern and form a cyst instead. Now, this has been mentioned a lot, the cilium. So what is the cilium? A cilia is an organelle, so it's a part of a cell, and it is present on almost all mammalian cells, so in all cells of our body. It's immotile because there is a motile cilia as well, which is present in the lungs, and it varies in length. So it is anywhere between 2 to 3 micrometers to 4 micrometers. And these numbers are all microscopic, so you can't see it with naked eye if you were to look for it. And what does it do? It is involved in signaling. So cells talk to each other and their own environment with these cilia. And they're very dynamic structures. The primary cilia, you can see on the video here, is a dynamic structure where there is movement of molecules up and down the cilium. And the cilia on the right is the static cilia. You can see that it has a ciliary membrane on the outside and this middle elevator-like structure in the middle, which helps in the communication. And over the past 2 decades, the cilia have really come to the forefront in studying a variety of different conditions. And a phenotype is how a disease presents itself. So we will limit ourselves to the cystic kidneys today. And cilia have been at the real heart of study for the cyst formation itself. And we, in the past sort of 6 years, have pioneered a technique in the UK, which I learned from our collaborators in Utrecht. Now, in this technique, we take the human urine, we take it off to the lab, and on the extreme right, you can see that these cells are growing in dishes, and these cells are basically kidney cells. I'll exemplify this a bit better in the next slide here. You can see the kidney. If you narrow it down, you can see the tubule. This is the functioning, beating heart of the kidney, the nephron. And within the nephron, there are these tube-like structures, and within them are these primary cilia. And these cilia are constantly being excreted by all of us pretty much all the time in our urine. And we now have the ability to culture them in a lab environment and test them for abnormalities and use treatments if we find any abnormality. This is how these cilia look under an immunofluorescence microscope. And we're calling it a liquid kidney biopsy because as some of you may know, a kidney biopsy is an invasive procedure and it's associated with a lot of risks. But collecting urine is a very safe, it's a replicable technique. It can be done really anywhere. And we are using this technique to culture these cells. The human urine-derived kidney cell look something like this. So just to orientate people who don't know this picture, the blue is a nucleus of a cell, and the green long bit is the cilia. You can also see on the right-hand panel that we have the ability to grow these cells in 3 dimensions, not only 2 dimensions. And you can see a beautiful lumen forming here and the cilia projecting on the inside. As I said, we've been studying the ciliary phenotype, so how the cilia look in a variety of conditions. And to orientate you with this panel, at the very top is WT, which is wild type, so normal cilia. You can see the green cilia quite beautifully projecting from the cell. In autosomal recessive polycystic kidney disease, you can see that the cilia are very similar to a wild type cilia. A condition called orofacial digital syndrome, you can see that the cilia carry a bleb at the very top. And the next 2 slides, we've studied this condition, nephronophthisis, in great detail over the past nearly a decade. And we found that in this condition, the cilia were very long and dysfunctional. So we asked the question as to what happens to the cilia in autosomal dominant polycystic kidney disease. Now, studying the human-derived cells hadn't been done before, and we started asking this question and looked at the cells which are coming down the urine of patients with polycystic kidney disease. And we saw that the cells carry a very long cilia. On the left-hand panel, you can see that this is a control cilia, which is short and normal. And in ADPKD, it's quite long. Now, this is ongoing work, so we are still making sure that our findings are replicable and we can see it consistently. The graph on the right-hand side shows that the cilia are about 8 to 10 micrometer in length in polycystic kidney disease, and normal cilia are about 4 to 5 micrometer. Now, we started then looking for these cilia under electron microscopy. This is another way of studying the cilia and the kidney. Now, again, to orientate you with this picture, this is the cyst within a polycystic kidney disease of a patient who had kindly donated their kidney after it was removed for research. And we took it away to the electron microscopy lab and studied the cilia. So this image is of the inside of a cyst. And when you get closer to the cyst with an electron microscope, you can see that these tiny hair-like structures begin to make their appearance. And when we zoom in closer, you can see that the cilia are quite long. In this particular instance, it was about 6.5 micrometer long. And our work is ongoing in correlating these images with individual patients. And in this panel, you can see that individual patients who are in our clinic, we are trying to make sure that we've got all the details possible for their clinical phenotype. So this is their clinical phenotype, whether or not they are on tolvaptan, what is the size of their kidneys, what is their kidney function, and what is their risk score of progression. And we are trying to correlate this with the Cilia structure. Now, to conclude about this part of my talk, we are trying to collate clinical data on patients with inherited kidney conditions, and this collaboration is going on between both mine and John's clinics north and south of the time. And the study of ADPKD, ciliary phenotype, has never been explored before, and we are pioneering this. And we aim to be able to establish another readout for assessing what is traditionally been based only on scanning and blood tests. And the impact of this work on the patients, we have established a very safe way of culturing kidney cells in Newcastle. And this has really expanded in more ways than one in our ability to investigate this condition. We have been collecting urine from patients across the Northeast, Yorkshire and Scotland. And we have identified various new pathways and ability to genetically manipulate in a slightly different condition from ADPKD. Now, what else is there on, out there in the on the horizon? Sorry, I moved on there. There are a fair few trials which are in at the moment phase 3. Basically, there are 3 phases of any trial. The third phase, phase 3 trials are human trials. So the first one is this lanreotide. It's called the LIPS study, and it's now finished, and we are waiting for the results of this study. There are quite a few exciting new molecules which are out there. Lixivaptan, which is similar to tolvaptan, more specific molecule. The tesevatinib, which is again an EGFR inhibitor, which is basically reduces the proliferation of cells within the cyst, and bardozolone is another molecule, and a small molecule at the very end, which is coming out in the study is finishing in 2023. So over the next 5 years, we are going to see results coming out from these clinical trials, which are currently ongoing across the globe. Now, this work, which has been very generously funded by the PKD Charity, I have a lot of people to thank to here, and the most important, the patients and their families. Kidney Research UK and PKD Charity have generously funded our work. Similarly, the European Renal Association and EDTA, Newcastle University, and John, who is here with us. A special mention to 2 medical students here, Rio and Tiffany, who have done some really excellent work in getting the clinical data together for our patient group. And I'll thank everyone here who are listening, and we'll be open to questions after this. Thanks.
Thank you, Shalabh. A couple of questions. Can I sort of raise this one to start with? So this is a question about cilia. Mm-hmm. And this person is asking, are there similar observations in liver cysts?
So we don't do any work within the liver. The liver cysts do contain cilia, but I don't think anybody has studied it as far as I know. I don't know, John.
No, so the cells from the liver are much harder to study. There are a lot less cell lines available, and obviously a urine sample wouldn't contain any liver cells. People can take stem cells and make them into liver cells, but that's very sort of labor-intensive way to study the liver cells. So yeah, much more is known about kidney cysts than liver cysts at the moment.
Thanks, John. Another question here. Is there any research around replacing the faulty genetic chromosome during the fertility stage to prevent passing the genes on?
Okay, I'll answer that one. So PKD is on the list for pre-implantation genetic diagnosis. So if you know your mutation in your family and you want to avoid passing that on to a child, then it's a process of IVF and selecting out the embryos that don't carry the genetic change. There isn't any gene replacement therapy at the moment for this particular gene. As you might know, PKD1 gene is huge. It's a very long, complicated gene with lots and lots of different parts. And to do a gene therapy that would replace that gene would be technically very, very challenging. So gene therapies are around. They're used for bone marrow conditions mainly, and venturing out into other conditions, but they're starting with the smaller genes that are easily replaceable. And I think PKD1 would be well down on the list of genes thought to be replaceable at the moment. But you know, anything's possible, and this field of research is moving quite rapidly. So it may come to the fact that we can start replacing genetic mistakes, but there's certainly no therapies at the moment doing that.
Thanks, John. And of course, we've got a talk on genetics coming up shortly, so that there may be more information during that one. I think, Tess, I believe you had there was an earlier question that you wanted to put to John or Shalabh.
Yeah, that's correct. We had an earlier question about naringenin, which was researched in 2014 in vitro and in animals. It slowed growth, cyst growth, but not cyst development. I believe it's a compound that can be derived from citrus fruit. And at the time, there were the usual headline in the tabloids: citrus fruits can stop PKD, which we come to expect quite often, and that of course caused confusion because a lot of patients can't, for example, eat grapefruits or sort of oranges, which interact with the medication. So that was a kind of flurry of interest at the time. To my knowledge, there's been no further work on it. It's a readily available product, and I think that's one of the problems with this type of research. As we know, there's research going on into turmeric and other ingredients and products that are cheap and readily available, but therefore of no commercial interest to the pharmaceutical business. So I don't know if, John, you've heard anything more about that one since 2014.
No, it seems to have gone quiet. But what I can say is, you know, the system that Shalabh has described, that we grow a cell from a patient, that cell has that specific mutation, and that specific mutation may respond to a therapy that's different from the next mutation. And so what we're doing here is a very personalized medicine approach. And so that I think we will find that some patient cells will respond to therapy A, whereas some won't, and then some will respond to therapy B. So I think the future of genetic diseases, particularly cystic kidney diseases, is this personalized medicine approach that, you know, it's slightly naive to expect one drug will cure everyone's version of cystic kidney disease. And so hopefully we'll derive therapies that we know work for a certain set of mutations, but don't work for the other set. And so, you know, we can be very clever and specific about therapies. Regarding that citrus supplement, I don't think there's any human data to support it at the moment. So I can't comment further on that.
Thanks, John. We've got a question here about ketosis. Before I go into that, I just wanted to sort of flag that we appear to have somebody in the audience with a royal title who wants to let you know that their liver is available at the tissue bank at Birmingham University, and you are welcome to it.
Okay, so I assume that's a cystic liver that's been removed because of its size, but that's very helpful. And these tissue banks are extremely important. because, you know, these samples are very, very precious. And you're right, we only gain knowledge by looking at, you know, microscopic level and, you know, cellular, molecular level, what's going on. So thanks for that information.
Yes, thank you. That's very generous. So the question on ketosis is, so this person's read an article about the research on the impact of ketosis in ADPKD. Would you like to comment on that?
Do you want to answer this one, Shalabh? I think you're on mute, Shalabh.
Okay, sorry, I've unmuted it now. We have to be careful when we read, you know, certain articles, you know, especially investigating one particular aspect like ketosis. Again, it's a multifactorial disease in a way that it progresses. So what we need to be very cautious of when we're reading article on one particular intervention is really take it in light of what the kind of patient was, where we are with their disease, and whether that would be the right approach for everyone. It would be my take on it. I have to say, I wouldn't give a one treatment fits, you know, everybody approach for this condition. I really tailor my treatments for individual patients in this one.
Thank you, and that's really useful. And I think early this year we did a webinar on ketosis by Thomas Vimes, and I'm sure he would mirror exactly what your advice is there, Shalabh. That's available on our on the PKD Charity website. We can actually send out the link after the event if anybody's interested in viewing that. We do have another question here about can study of cilia help the, I'm sorry, I can't pronounce the word, it's
prognosticate.
Thank you. Thanks, John. Perhaps you can answer that then.
Yeah, so I think the question's meaning, does an abnormal cilia tell you what's going to happen in the future, how severe the disease is going to be? We do think that we can learn this information. So as Shalabh was saying, in a related disease called nephrolithiasis, we can take cells from the urine and grow them, and we know that they predict disease severity in the kidney. So we'd like to continue the work in ADPKD to say, okay, let's put these things together. The patient's genotype, that's what genetic change they have, their ciliary phenotype, i.e. what the cilia look like under the microscope, and then their clinical picture. And I think we'll be able to tie that information together to say, okay, you're at low risk of getting kidney failure in the future, or you're at medium risk, or you're at high risk. And so I think the cilia can help us to prognosticate ADPKD, but this is all work in progress. And, you know, as you know, nearly everyone has a slightly different mutation in the gene. So we want to be really sure that we're not making generalizations, and we want to know, you know, does a truncating or a stop codon mutation give different cilia to a missense mutation? So, you know, these are the studies that are all ongoing, and each urine sample that we get for anyone from PKD is very, very precious. The other thing that we're trying to work out is, what does tolvaptan do to the cilia? No one knows. And does tolvaptan rescue a ciliary phenotype that then allows you to gain kidney function? And no one knows that question. I think it's quite an interesting question.
Oh, sorry, Jane. John, a sort of follow-up question to that, really. What therapies are you proposing, therefore, or looking at in the future to correct the cilia, whether it's too long, too short, got a blob on it, etc.?
Yeah, so as you all probably know, there's, you know, you can draw a cell that's affected by PKD, and there's 30 different mechanisms and 30 different treatments to act on all those different pathways. But the work that we are, you know, pioneering, actually, that Shalabh's pioneering, is specific genetic therapies to actually either correct that genetic change on an individual basis or allow the cell to ignore the genetic change. And so we think these personalized sort of genetic therapies are one of the ways forward that avoids, you know, toxic effects of drugs, side effects of drugs, because we know these therapies have been used in different conditions like muscular dystrophy and other genetic disorders and have very good effects without side effects.
Thanks, John. There's just one question here, and I think if I understand this correctly, I think that this person may be asking if the studies that have been conducted so far show if PKD is related to any other syndromes. I may be completely misunderstanding that question, but maybe you can sort of shed some light on that if it's relevant.
Yes, so shall I take that one?
Yeah.
So I think what we have found more recently is that there are not only PKD1 and PKD2 genes, there are other genes now that have been found in the last few years that have shown similar sort of cystic pattern within the kidneys, but they also come with other related conditions. So you could have aneurysmal dilatation of some blood vessels. You can also have other related conditions. So I think, yes, the cilia will give us an idea. But again, as we've been saying over and over throughout this talk, is that we have to correlate the phenotype, as in the disease, how it is presenting, and then the genotype, and put the two together to find newer information. And that is why we have now, embarked upon a really deep description of all our patients. And that's why I had thanked the medical students who've been helping us, because we've really been looking very carefully through every individual patient as to how they presented, what was their family history, were there any other conditions which have been sort of running in the same pattern, and then study their cilia, and then put the whole information together. So yes, to that question, yes, it may tell us that there are other syndromes or just another way that this condition presents, not just the traditional description, which is the PKD1 and 2 mutation. John?
Yeah, I'd agree with that. And we do know that, we're calling PKD a ciliopathy these days. That's the term because the cilia are wrong, and alongside the cystic kidney disease comes other things like cysts in other organs and blood vessel problems. And these are all expected because this is where the PKD proteins are expressed.
Thank you. Well, just one final question before we go on to our next speaker. This person is saying that they've seen some research that suggests that Usher syndrome may be connected to PKD in some way. Could either of you comment on that?
Yes, so Usher syndrome is what you'd call a ciliopathy, and it typically causes deafness and some eye problems and can lead to blindness. This condition called RP, which is retinitis pigmentosa. And so Usher would be, a typical ciliopathy, and that you can get extra manifestations, bone, joint problems, cysts in other organs. So this is all part of, the picture of some of these rarer conditions. So Usher syndrome is a much rarer condition than PKD. We work on a condition called Joubert syndrome, which again causes brain and eye and kidney problems. And so ciliopathies are this sort of umbrella term to capture all these much rarer conditions. And so, we are making connections between the rare and common forms of ciliopathies all the time. So, yeah, on a case-by-case basis, there may be some interesting insights into how Usher syndrome is connected with cystic kidney disease.
Yeah, I mean, if this person is saying that their sister has RP, whereas they have PKD. And I guess, you know, that familial sort of connection is there something in that one has PKD, the other one doesn't, but has RP?
Yeah. So, you know, I can't comment on the personal case perhaps, but, you know, one has to be very clear that the cysts you have are from PKD1 or PKD2, and you know, if they're not, there could be a more unusual or exotic form of your cystic kidney disease, and therefore, you know, genetic studies are the only way to sort of sort you and your sister out in terms of what's actually going wrong. So things can look like PKD, but can be a rarer form. And as Shalabh said, you know, in the last 2 years, there's maybe another 5 genes that have been identified that can cause things that look just like PKD1 and 2, but are slightly different versions of it and come with slightly different other effects on the body.
Thanks, John, and thanks, Shalabh. We've come to the end of that now. We're ready to move on to our next talk. But thank you very much
Questions.
for attending. Thank you, everybody, for all your questions. There was some interesting questions there.